Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Quick note before we begin, the Finding Genius Foundation, as part of the Finding Genius Podcast, has recently completed a book about understanding viruses. So the creation of this book was to interview 100 virologists, ask them a lot of deep, difficult questions, take the most difficult questions, and then re-interview the top 25 or so and ask them the hardest questions I could think of. And we compiled that all into a book. So you'll see question and 4 or 5 experts' answers. Question, 4 or 5 experts' answers. There's about 30 questions in the book. I think it's a great read for the layperson and for the researcher. It talks about a lot of speculation in the world of viruses, such as are they alive or not, and why is it important? Why do viruses go latent or hidden or ineffective or sit in a person or an animal or another creature for weeks, months, years, and then suddenly become virulent and affect that person? So there's a lot of really provocative questions in the book. It's now on Amazon. So if you go to Amazon and type in Finding Genius, you'll see the book on viruses. It's also on Kindle. The Audible version is in production and should be ready in approximately a month. But if you want to go and order it now, you can do so again by going to Amazon or Kindle, or go to findinggeniusfoundation.org and go to publications. There's an opportunity as well to get the transcripts of all the interviews and to hear the original interviews themselves. If we put them all together, the book would be about 1,000 pages, but we condensed them down to make it juicy and concise and tight and very interesting. So I hope you'll check out the book. We're now working on one about cancer, but this is going to be our goal is 3 times a year to come out with these masterclass books that I think will inspire new scientific research, and I hope you'll check it out. Thank you.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. My guest today is helping me out as a co-author of the cancer book I'm putting together. It's Dr. Steve Gullans. He has broad experience as a consultant across the life sciences industry. He was a board director at Gemfire starting 2016. He's also was a managing director at Excel Venture Management, which was a Boston-based life sciences venture capital firm that he co-founded in 2008. So he's been around, had a lot of experience, and we had a great call last time, and I wanted to have him back for the cancer book. So, Steve, thanks for coming back.
Thank you, Richard. Great to connect again.
All right, so my first question is, do you think or perceive that cancer is essentially a separate life form inside of us? And if so, when is that? Is it when it's a few cells, when it's a billion cells, when it's metastasized, or is it never a life form, and is it something else?
I don't view it as a separate life form in this for a number of reasons. It inherently is dependent on, it's not that you can't be symbiotic and not be a separate life form, but in fact, it works in synchrony with our body. It is not propagating itself into more life forms. It does not reproduce except within your body. So I see it as a standalone entity, a separate organ, if you want, in our own bodies with its own control system.
Do you think it has its own homeostatic drive? Like, if I have a primary tumor, does it act as one? Does the tumor have cell-to-cell signaling and, saying, I have a hypoxic region, let's get some blood vessels in here? Or do you think that's just individual cell needs?
I think every cell's responding as a single entity. I was part of the team that sequenced the first solid tumor ever in history. And we actually, some of the data did not end up in the paper, showing all the different subtypes of lineages that occur with different genetic mutations. That subsequently came out as information that's out there. So just like having great-grandparents and then progeny and progeny below that, you're seeing changes that go on, but no cell stands in isolation. It has to beget sister and daughter cells that it can live with. But in the end, it's a community. It's a community that lives together. And it's responding to the world around it, which is an inherent part of Darwinian evolution, responding through natural selection to environmental forces around you.
Okay.
That response can be a community of slightly different cell types. It doesn't need to be a single cell type.
Okay. Do you think that there's any communication between a primary tumor and metastases? Like where the primary would be the boss that tells the metastases what to do, or is there any coordination at all of action?
There's no coordination of action. The mechanisms are pretty well known for what causes a cell to metastasize. If you think about it, these mechanisms are inherent in all the other basic normal functions in our body. We have bone marrow cells that actually release from bone marrow that are stem cells that go to other body parts and become other cell types. And so the idea of homing into a new place in the body and functioning in that case, you can even change its phenotype or its true type of existence to the local environment. So I think it's just the idea of metastasizing as a natural element of the body, particularly among the lymphocytic and the blood cells of our body.
Well, why would certain cancers have tropisms for certain areas, places they seem to prefer to go?
Well, part of it is just simple blood supply. And the other obvious part is that they have to escape from the blood vessels if the blood vessels are carrying the cells. And so there are some places that are easy to get into, some are more difficult. And then there's the actual communication system for when a cell migrates into a tissue, it has to bind to the wall of the artery or the vein and migrate through it. And so all of those steps have very specific mechanisms, no different than you can imagine for attacking a virus. It was in that region in a specialized cell type. So there are going to be natural preferences think of it as a cell from your bone marrow that was a cancer having a key to certain locks that are in certain tissues. It's always going to find those tissues. We have not understood that completely. And obviously a part of it is it's a very dynamic system that's changing very quickly. But there are natural alliances when certain kinds of tumors end up in obvious places you go looking in terms of the migration. But there is no direct communication between them, aside from just general factors that get secreted to divide and grow and other things to the whole body.
Okay. You mentioned that you were one of the first to sequence a solid tumor. Yeah. Now that there's single cell sequencing, has anyone, let's say they had a spheroid type tumor and they resected it, yep, and they sequenced the heck out of it and they tried to see where the mutations are spatially and then maybe model it and go backwards in time and see how it first began or how it grew. Is that possible yet? You can even do it with just simple computer algorithms, because you look for the percentage. When we first did this, we would see that you could do it just by counting the number of chromosomes or which chromosomes had duplicated. And you would say, oh my goodness, a piece of the Y chromosome has suddenly duplicated in this portion of the liver where there's a tumor. And so all of those cells came from that a single event of that partial Y duplication. And so you could actually reconstitute it at X percentage, because it's the chromosomal abnormality. It's not true in every tumor, but you can actually recapitulate it informatically. So that's the power of single cell sequencing, and the informatics is beginning to put together this dynamic understanding of that every cell is different in some way, but the difference disappears in the aggregate when you look across the whole colony. The largest difference when you're doing single cell sequence, especially in tumors, is the presence or absence of different immune systems. Immune cells. So when you have a hot tumor filled with lymphocytes, the immuno-oncology drugs like Keytruda and Opdivo are working because they actually have a way, those cells are already there and can be recruited. Whereas cold tumor had none of those lymphocytes around in it. They're all around it. And so that's the largest single cell observations that have come about. Recently with COVID, there was a paper that came out today in Nature saying that macrophages, monocytes, are the most invasive cells that are the culprits in the lung injury that people die of. So you're actually starting to see which immune cells are the culprits in any of these disease settings. So what do you observe by looking at the placement, let's say, of different mutations in a cancer? Does a cancer look like a Jackson Pollock, or is it like a Picasso where there's areas of organization, but they're kind of jumbled? Or does it look orderly at all, or is it just a complete mess?
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit FindingGeniusPodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit FindingGeniusPodcast.com and click Support Us today. Now back to the show.
I think it's a mix of all. There's always a few what they are called driver mutations, and there's maybe 50 genes. That P53 would be one. There's a few genes that show up repeatedly because those are a fundamental mutation that is part of a tumor. But a single mutation is generally not sufficient for the immune system, for the tumor to grow and escape the immune system's attacks. And so you'll see sublineages with, say, P53 is the key one, or RAS or MYC or a lot of these other oncogenes are the key mutation. But then you see submutations that are adapting to the environment, whether it's hypoxia or the individuals are diabetic and has glucose pulses. You're going to see subpopulations that are intuitively grow faster. The strongest agent driving modifications is all the interventions we do therapeutically. Doesn't matter if it's chemo or radiation, the cells will respond. They'll find a way, hopefully, first to survive and then to proliferate. And those are big drivers of moving into new areas of new mutations and new cell types, new phenotypes.
Primary tumors, their microenvironment is still, cells that they came from, but metastases are in a more alien microenvironment. How do you think that affects their ability to evade the immune system and to function and to grow and to proliferate?
That, not sure about it. A lot of it's going to be very specific to the tumor and the microenvironment. There are certain elements of the microenvironment that are generic, certain fibroblastic parameters, certain ability to bring in blood vessels and to grow that all tumors need. But within that milieu, there is the immune system that may or may not be an active participant in the cell tissue type to begin with. In addition, you'll see local hormone influences that may or may not be active. But I don't think there's enough information to answer all the aspects of metastases. In general, we've treated metastases as if they're the same as the primary. And we see the efficacy of therapeutics is very poor in metastatic tumors. So undoubtedly, there's something different. And we just haven't done the groundwork, nor have we had a chance to try alternative therapies, because that first line works in the primary tumor. Let's try it first in the metastatic tumor. It's going to take more time and more different kinds of therapeutics to get at that.
If I looked at a couple of different tumors, let's say a big tumor in someone's colon, there's a lot of microbes there. It's a microbiome. I would think that each cell, because it's mutated differently, would have different metabolic needs slightly and would attract maybe different microbial players. So what would you expect to see if you were able to somehow hyper-localize the microbes that were around each cell in a tumor? Would you expect them to be different? And what do you think that would do? How would that affect the whole mass?
Well, not every cell is mutated completely differently. You're going to find large domains or clusters of cells that are very similar. The bacteria are even more opportunistic than cancer in adapting to their environment. There's many, many more species of bacteria and even more viruses living in our gut that can be, particularly in the digestive tract, are a part of the microenvironment around a tumor. And they're activating the immune system itself. So the microenvironment that exists between the microbes in the tumor is an area currently of active investigation. The first pass at this kind of investigation is simply trying to find correlations for this kind of tumor. And you can characterize it as a cell type or a drug responsiveness or whatever it is, has more likely is going to see these kinds of bugs or microorganisms around it. We assume that it's because the microbes can survive there, but there's also this obvious line of thought that they're actually supporting the tumor. A lot of microbes are very good at making short-chain fatty acids, which are a part of the treatment of epilepsy, for example. So they clearly are benefits for microbes that make certain types of foods. The tumor can adapt The microbes can adapt, but there's a symbiotic opportunity there. And whether it actually pans out, especially over time as the tumor is growing and the environment's changing, is unsure.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
And the other piece of it, obviously, is it's an inexact experiment because the diet, especially for people on chemo, people taking supplements of cancer patients, varies over the course of the disease because as a human being, you're trying to adapt to live the best possible quality of life that you can. So we will mostly see correlative studies for quite a while. The idea of engineering bacteria to not deliver, say, a fatty acid that this tumor likes is years away, but technically within our domain right now. We just have to have good rationale for doing it.
Do you know anyone that's looked at the microbiome of people that have various cancers and, contrast it with, if you were, this would be hard to do. Ideally, it would be nice if everyone, let's say, got their microbiome sequenced every 6 months. And then if they get sick, you can compare how it would change. I don't know how much that would tell you, but
right.
Do you know anyone that's looking at this?
I know people have done it in mice because it's a single tumor type. You implant an identical tumor to all of them. So it's not a different tumor environment. You can give them all the same diet. So you start to see that's where the correlations begin to become more obvious. The idea of doing it in humans is challenging right now, just for, not technically, but just in terms of interpretation. It would be very difficult in today's world of mechanism to plausibly just state you have a mechanism and get it published in a high-quality journal. I'm sure people are doing it. We're just not seeing those papers getting published, that if you have this tumor, these bacteria are good, and those are bad, is not even been brought to a level where people believe it's true.
Yeah, it's very complicated. I know there's a long way to go there.
But it's the problem is fundamentally it's not about experimenting on humans. We just can't. There's such diversity among how all the humans get the disease, treat the disease, and everything else, that when you have an external factor like microbes that are based on the diet you eat, the antibiotics you take, all the drugs you're on, it's very hard to get a single answer. What
about liquid biopsies or alternative biopsy methods, especially for hard to reach cancers, pancreatic cancer, et cetera? What's some of the new innovation there?
Well, there's lots of great innovation on the diagnostic front, and we could talk a little bit about that, where it goes therapeutically. The liquid biopsies arose basically from the surgical suite, realizing they could get a piece of the tumor, at a small piece of it, and begin to do things with it. And it was very informative, and I was actually involved in some of those early studies. We were starting to see fundamentally differences in the presence or absence of immune cells. What came after that was the idea of testing your blood for DNA, this characteristic, or even your stool with the test from Exact Sciences, looking for DNA clues for tumors, really looking for relatively frequent mutations that are like in RAS or P53, or just looking for too much of a particular chromosome. There's other markers that you could actually see. But at the end of the day, what you care about is statistically on the therapeutics, true positives and true negatives. I had one senior executive tell me until you get above 95%, you don't want to have people going in for biopsies when you're guessing where the tumor might be. And even if you do fine needle aspirates or fine needle biopsies, what do you do with the data that changes your treatment paradigm? And that's the mismatch. For all human diseases, we always have been able to diagnose before we therapeutically can address. Dr. Alzheimer's first discovered Alzheimer's in the 1800s. Sickle cell anemia, so the mutation was found in the 1950s, and we're just now talking about treating it. So in cancer, it's not that different, except that we're on a much more rapid trajectory. Our ability to find, detect, and even identify causal mutations of tumors is vastly exceeding the number of drugs we have. So it's as if, imagine there was 100 mutations that needed 100 drugs. Well, we would only have 3 to choose from. And that's why there's a mismatch. But with the informatics opportunities and some other new technologies that are making things in vitro better, we will begin to change that landscape. There's still a number of hurdles. And, we could talk about those if you like, to getting this whole paradigm of define the molecular phenotype and genotype of a tumor, and then define the course, the trajectory, and the prognosis for a patient going forward, and how we actually address that quickly.
Yeah, how can anyone tell based on after they biopsy a tumor and they look at all the mutations, or can we tell which direction it will go in, how aggressive it'll be, how it'll respond to chemo, et cetera?
Right. Well, there's a couple of rules of thumb and a couple of barriers to advancing things. One is that one thing that's been a false lead is penicillin. We got the impression that infectious disease, that a single pill, a single drug was sufficient to wipe out a disease, or the vast majority of it. And that worked in infectious diseases and a few other isolated examples. Why that was so successful was, first of all, nature had figured out how to make these things, bugs killing each other. But more importantly, you're actually killing an organism that is genomically and fundamentally different than the human body. So toxicity is not such a big problem. You know you're going to kill it if you get enough of that stuff in your body. And with a tumor, you don't know that going into the experiment. So we were misled by that. The other thing is just when you do get a single drug, even in an infectious disease like HIV, we saw the ability to mutate around it is unbelievable in the case of a virus. But it's for human cells. If you do things serially, you do drug at a time, inevitably you're leaving opportunities and selective pressures. So the idea of double, triple therapy becomes very obvious. And the question is, you now have an exponentially growing number of drugs you should try together to see what works. So how are we going to disentangle that web of potential drugs that work? Well, a couple of thoughts around that. One is there are experiments being done, particularly in the organs on chips scenario, where the efficacy of having real human disease, say tumors growing in plastic chips with blood supply and everything else, is beginning to recapitulate the efficacy that we see with drugs in humans. The FDA is backing a lot of those studies. You're not testing in mice, you're testing real human disease cells. So you can actually start to do combinations in a time and cost that's reasonable. The other is when you begin to pile together more than one good drug, if it's not just the generics, the cost of manufacturing, the cost of bringing it to market is so high that we now support, $10, $20, $100,000 for a single drug. If you put two together, you're doubling that. You put three together, you're tripling it. The cost of manufacturing and getting to drug through the pipeline is long and hard. That has to change too. The good news is we've seen it change with the vaccines that came to market in the past 12 months. It's not that it's not possible. It's just there's a few factors. One is, while these were high-risk efforts with the mRNAs, their efficacy appears to be unbelievable. So if we go from the standard efficacy of a drug coming through clinical trials in cancer, which is 5%, and then look at all the vaccines, even the ones that didn't do very well at 50% and were abandoned, the efficacy was unbelievably high with these new technologies because they were actually targeted to the specific thing you were looking at. And so we can actually have higher efficiencies in efficacy. Toxicity we have to work at a little bit more in terms of making better predictions. But the third thing is just the simple manufacturing, the millions and millions of dollars you spend, and nobody ever takes that risk ahead of finishing their trials, because why would you waste your money? I know the Gates Foundations and others are looking for, this is sort of the last hurdle to get these truly revolutionary therapeutics into humans, is let's reduce the time and cost of going from, I have a great drug I want to try in real patients, from 3 years and say $10 to $20 million just to get to the patients, or maybe 2 years, down to a few months, and with small incubators or, flow through incubators that can do it on the fly. If we can actually make that transition, everything comes down 10x. We begin to think about these multiple cocktails of therapeutics and marrying the true diagnostic markers, the diagnostic with different regimens, and trying them.
Does the way the FDA approves drugs, though, does it support that kind of paradigm, or will the FDA have to change how they do things, and are they willing? Are they listening?
The answer is yes, and probably. The yes part is the FDA has actually been out front trying to bring new innovation into the industry. They actually look at some of these technologies, adaptive clinical trials, organs on chips, all of these things. And with the vaccines that came, they were not an impediment. They really were not an impediment, despite what you might pick up from the media. They were on board for all the obvious reasons, with the bar being you cannot kill a single person, which is a pretty important bar, except that in the timeline they're going. So they are being encouraged by, frankly, all administrations to date, and even some of the new heads, to be more liberal in their thinking about how do we get things there faster. The industry has been a little reluctant to, because people who are in the business of toxicology, in the business of manufacturing, only lose their jobs when they make a mistake. They don't like to change policies or plans. Say, you know what, if we use this gene chip technology to figure toxicology, we'll save a year. Well, what if it fails? They lose their job. So there is a risk in adopting new technologies. But now that we've seen it work once, and we've also seen a global community involved in all of these activities, and all of the recipes to make these things, do these things, are in the patent literature. So you will start to see elements of this appearing everywhere else around the world, and the FDA will want to keep up. So I think change is always much slower than you expect. But as we get our efficiency up, which has doubled in the last 7 years of drugs coming through the FDA pipeline more frequently with success, people are going to be more permissive about what we need to do to get approval.
Does the FDA require currently that any change you make to a cocktail, or that all elements of the cocktail are tested exactly in a certain proportion or individually?
Yeah, they have very strict rules around that. The quality of the materials, what the proportions are, and the assays that go into them. And that's one of the hurdles to, you would prefer to have everything in a single cocktail as a way to protect that. But if you're taking different mixes from different places, you're not always sure. It adds another level of complexity to the synthesis. Anytime you mix two drugs into a single pill, there is a technical as well as a regulatory hurdle you have to go over that doesn't exist if you take two separate pills.
So what do you see as the future of cancer treatment? Is it going to come from drugs? Is it going to come from more chemo? Is it going to come from a better understanding of how cancer functions? I mean, where do you think it's going to be?
Well, the human genome was done about 20 years ago. At that point, we had the entire recipe book, but we didn't have the recipe. We had all the ingredients, all right? We didn't know the proper mix. For 20 years, we've been doing a few things, whether it's small drugs or genomics or drug discovery, biologics, is we've been trying to figure out what the recipes are. And because we've been constrained by really testing one ingredient at a time, it's been a challenge to make huge strides in cancer. The success rate of 5% is very low. The average across all drugs is 9%. At the same time, there's over 7,000 clinical trials ongoing for cancer. The next closest is 1,000 in infectious diseases. And so we have enormous amounts of opportunity to try to tackle this. What we're seeing is that there's fundamentally a disconnect between toxicity and efficacy related to making the drug work only in the tumor cell. If the drug could target only the tumor environment, we have probably 50 drugs, biologics today that would work. Interferon, interleukin, you name it. If it only went to the tumor. They were all tried in the 90s. There's over 2,100 interferon gamma clinical trials, and almost nothing's on the market that's very toxic. You see, I've seen multiple technologies that are starting to target. There were first attempts to do this, but started to target nature's own cocktails of what kills tumor. Think about something like tumor necrosis factor. Its name tells you what it does. But if you inject it into a human, they're dead in 2 hours. But if you inject it just into a tumor at the end of your finger and collect the TNF that comes back, you get a 100% cure rate. So it's partly about delivering to the cell type of interest. The other is actually the riddle of which therapy matches which disease or which tumor type. Well, we need to solve that first step first. When we do that, we can start to mix and match a little more. And that fundamentally comes back to what I was saying before about the cost of making these things. As they come off patent, and fundamentally as we get cheaper ways to make small batches that could be deployed to small trials, I think we could actually see a lot more opportunity going on. One of the things I find very encouraging is for 40 years, from the 80s to actually for probably 60 or 70 years, from the 50s to about 5 years ago, the average was 24 drugs a year get approved by the FDA total. That includes Me Too drugs. 6 years ago, that number changed, and we're now up above 50 drugs a year getting approved. That and the number of trials that have been started per each year has not changed in the last 10 years. So our efficiency across all diseases has doubled. The efficiency in infectious disease is 25%. But here we had all of these new vaccines come through with 80% success rates. And most of the time, we still don't have a vaccine to HIV or to malaria or to many other things. So we are figuring out how to build these cassettes, what to put in them and how to make them work. So the efficiency is going to drive it because it will drive more money in, it will bring more people. People will be more willing to take the toxicity risk. It's more rare and the likelihood of success is higher. And finally, the part that I see a lot of in my day-to-day activities is in the last 2 years, $26 billion of venture capital money has come in to the United States. New venture capital money has been dedicated to life science investments. 10 years ago, it was 1.8 in 1 year. And that does not include the public market. So God knows how we're going to get 2 to 4x multiples on 26 million over the last 2 years. But I hope we do, because the efficiency has to go up 2, 3, 4x. No longer should we have 24 drugs a year, 50 drugs a year where we are right now. But across all indications, with the lion's share of trials in cancer, we should be at 100, 200 successful outcomes a year.
Well, very good. Well, Steve, thank you again for coming back. What's the best way for people to find you and reach you now that you're doing full-time consulting?
Steve.Gullans@gmail.com. If you go to the internet, and you can look up my book, Evolving Ourselves, I wrote with Juan Enriquez. It's pretty easy to find me, or just go to LinkedIn.
Okay.
Thanks.
Well, very good. Steve, thank you for coming back. I appreciate it.
It's great to connect again, Richard. Thank you. Bye-bye.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.